Yeah. Yeah. Let's see. Hopefully it's gonna work. Yes, it did. Thank you. She, she's supposed to show up, yes. Should I, should I take a look if she's outside maybe? Looking for... Thank you. Yeah, I know. I have to go. Very good. Ah, she's not coming. She couldn't. Okay, thank you.
I checked from the long distance from the. I couldn't see her. Neither short or long. Maybe we get started. Should we talk?
Yeah, I think so.
Okay. So it's my pleasure to welcome you to this public thesis defense from Konstantinos Kompotis. Everybody knows everybody. I'm supposed to say a few words. He got a bachelor in biology in Greece, Thessaloniki, and a master in Netherlands, Maastricht. And then in 2014 joined Potsdam for a PhD. And also the neuroscience elementary program, which I represent for this final thesis defense. He went through a number of requirements, long series of requirements, and most importantly, private thesis defense with Professor London and Professor Witty as experts, with Wolfgang came with a long list and long series of questions, good discussion. And all these steps went very well. And that's why we are here today for the final step, then it will be open for another series of questions.
Thank you very much. So it's a very weird moment. Thank you all for being here. My presentation's title is Tuning Sleep, a molecular and a sensory approach in mice, meaning that I had two projects throughout these 5 years, and at least to me it seems like I was trying to do exactly that, to figure out in different levels how sleep can be optimized, let's say, regarding to the needs of the environment each time. So it definitely wouldn't be a sleep PhD defense if I didn't refer to this model. So this is the two-process model. This is how we think sleep works. Sleep is regulated by two processes, namely the process S, which you can see in the curve up here, and the process C. So the process S stands for sleep homeostasis. Homeostasis coming from the Greek word homeostasi, which is, in other words, balance, sleep balance. So again, in simpler words, the more you are awake, the more you need to sleep. And process C coming from standing for circadian rhythms, or in more simple words, what time of the day is it? Now, one other aspect that is very important for us is how we do basic research on sleep. And we do so by using models, and in this case, mice. So we put electrodes on the skull of mice, and we are able to record their brain waves. By doing so, we get something that looks like this. So we are able to assess whether a mouse is in non-REM sleep, REM sleep, or is awake by assessing basically the brain waves and as well as the electromyography, so the muscle movements. So as you can see here, a mouse can be in non-REM sleep, which is defined by large amplitude, slow waves. REM sleep, which is defined by short amplitude, very fast waves. And wakefulness with the analogous EMG, so muscle movement. Now, particularly when it comes to non-REM sleep, we have a way to assess, well, or let's say correlate, to assess the good quality of sleep. And this correlate, well, as electrophysiological correlate, is called EEG delta power. Now, what is this delta power? Through some mathematical equations that are not really necessary for this presentation, it's possible to assess exactly what the prevalence of these large waves, so high amplitude, slow waves, is. Basically what it means is the higher EEG delta power, the more of these slow waves, and therefore the deeper the sleep, the better the quality of the sleep. So just keep in mind that non-REM sleep EEG delta power reflects the homeostatic process. So if there is high delta, good sleep. If there is low delta, less good sleep. So it's possible to do sleep research at different levels. Starting from the molecular level, so what happens basically inside a cell. This is a neuron, by the way. Also at the level of communication between neurons, as well as at the level of brain structures, and even, so not exactly in this case brain structures, but also different organs, different systems of the brain. So in my case, what I did for the last 5 years was one project that focused on the molecular level, so the molecular regulation of sleep, and another that focused more on the higher level, so what happens between sensory organs, what is the effect of a certain sensory system on sleep. So these are exactly my two questions. The first chapter of my defense had to do with the molecular level, and more specifically the question was whether microRNAs, and a specific microRNA, is involved functionally in sleep. The second chapter, the question of the second chapter was whether rocking, the movement that we all know in babies or in cars, can actually, no, actually, we know that in humans it can affect sleep, but can it actually affect sleep in mice? And if so, how? So, let's begin with the first part of my thesis. The role of microRNA 709 in sleep regulation. Now, before I continue with microRNA 709, I have to explain to you what microRNAs are. And to do so, I have to go first through what DNA, so the genetic information being first in the form of DNA, then in the form of RNA, and in the end forming proteins. So, let me rephrase, please. Proteins are necessary for a cell to function. Now, the information to build a protein, such as an enzyme or a receptor, is actually coded in RNA, and before this level, it's coded in DNA. So this is the central dogma of biology. In this step, just before RNA becomes protein, there are several regulators. One of them are microRNAs. So what do? Basically, they exist in the cell. They find targets, their RNA targets, that are complementary to them. They bind to them, and they stop them from being translated into proteins. So it's good to know that microRNAs are short RNA molecules that do not code protein. They suppress the expression of their RNA targets, as we said, and one microRNA can actually target approximately 100 genes. So you can also think about them as the pedals of a piano. So they do not change anything from the notes, the actual notes of music, right? But they do modify according to the needs of the cell in this case. They do modify the end product, which is in this case the sound, in this case the protein. So what about microRNAs in sleep? There have been several studies about, well, not a lot though, several studies and very recent studies about microRNAs in sleep. Two of them were in Drosophila, another model, another two in the rats, and one in humans. Meaning that this is, although microRNAs have been known for around 24 years or so, no, exactly 24 years, they just started being, let's say, famous in the sleep field. So there's still a lot of room to cover. Now, previously in the lab, a previous PhD student found that when you sleep deprive mice for 6 hours, also several microRNAs are differentially regulated. So some microRNAs go up, some microRNAs go down. A certain specific microRNA 709, was the top candidate, was chosen out of these microRNAs as the top candidate. And its expression was actually, its upregulation after sleep deprivation was verified by in situ hybridization. So, what does this all mean? Basically, this is a brain slice from a control mouse, a non-sleep deprived mouse. The difference that I hope you can see, the difference in the signal, so the small blue dark dots here that are increased in this slice, means that 709 levels have been upregulated after 6 hours of sleep deprivation. Now, what else do we know about this microRNA? 709, we know from literature that it's enriched in neurons, and we also know that it's an activity-dependent microRNA, which makes sense also then that we see it being upregulated after 6 hours of certain activity. So, what was our question? We wanted to learn whether this microRNA 709, whether it's functionally evolved in sleep. What we did was to inject inhibitors of 709, either actually inhibitors or a negative control or a vehicle in the ventricles of mice. And we recorded the mice for 4 days, sleep depriving them as well after 2 days. So what would happen? What did we expect to happen in their brain? every cell that would absorb this inhibitor, this specific 709 inhibitor, would actually have the microRNA blocked. Therefore, the targets of the microRNA would be free to be upregulated. So, this is what we saw, trying to assess the EEG spectrum, so the brain waves, trying to assess the power from the different brain waves in the three groups. So, in green, the 709 inhibited mouse, in blue, the scrambled, the negative control, and in dark, the vehicle mice, vehicle injected mice. So we saw that basically, even at the last 4 hours of the light period, where mice have the lowest sleep pressure, there was already a difference at the range of the delta power. So remember, as we said before, delta power is a correlate of deep sleep. Now, this increase was actually larger after 12 hours, during the 12 hours of dark baseline, where mice are spontaneously awake. And lastly, we saw that this increase remained larger after the 6 hours of sleep deprivation. So what does that mean? That when you inhibit 709, delta power and a specific range of that delta power goes up. So we wanted to follow this band throughout the recordings. Here you see the average baseline, so 2 days of baseline averaged. You can see the delta power from the brain waves of the mouse. And I hope you can also see that the green group, so the group in green, which is the 709 inhibited group, is always above the other two overlapping groups. However, the difference in EEG delta power becomes significant only during the dark period, where mice are spontaneously awake, or after forced wakefulness, so after the 6 hours of sleep deprivation, which for us means that 709 might link what happens during wakefulness with the processes that happen during sleep, the recovery processes during sleep. Now, underneath, this panel shows only the fact that nothing changed in the times that they spend every hour in non-REM sleep. So it was solely in the brainwave, in the quality of their sleep, and not in the amount spent in sleep. So, okay, we did have a phenotype. Now the next question for us was, what is the mechanism underlying this? And to answer this question, we cultured primary cortical neurons and treated them exactly with the same inhibitors. So either a specific inhibitor for 709, a negative control, or an untreated control, the vehicle, basically. Now, we expected exactly the same to happen, that when we block 709, the targets, the specific targets of 709 should go up. And we assessed gene expression, so we assessed the levels of these transcripts by RNA sequencing. This is exactly what we saw. So first of all, 13,760 genes were identified by the RNA sequencing. Out of those, only 111 were differentially expressed. And here are some examples of these genes. Basically in red, you can see the genes that were significantly differentially regulated. Now, what was important was that most of the genes that were downregulated had to do with the electrical activity of the neuron, whereas the upregulated genes were mostly associated with the transport of proteins in the neuron. So, in this case, Paul asked a nice question. Basically, we have compared in the previous analysis individually the genes with each other. Is there a way where we can group genes and in their pathways and say something about these pathways? So what happened to the pathways that these genes participate to after 709 inhibition? And actually, this is the analysis that especially Jeff was waiting for. So there is a way, there is actually more than just one way. The weighted gene co-expression network analysis, or WGCNA, does exactly that. It identifies which genes in a given dataset, which genes are co-expressed, so they behave in the same way in all samples, and it clusters them into groups. And after that, you can find exactly which these groups are, what kind of genes are in these groups, and which pathways are enriched in these groups. So what I'm showing here is just an explanation of this. Each line is actually one gene. And below, these are the groups in color-coded groups that they have been clustered to. Now, on the right panel, what you can see is actually which of those groups was significantly different after the 709 inhibition. And what is also important to know is that these three groups were indeed different. And the first one had genes associated with neural and electrical activity, whereas the other two contain genes mostly associated with energy metabolism. Now, one other thing that is important is that these genes, the genes belonging to these groups, all of them were upregulated after 709. So after 709 inhibition, whereas these genes were downregulated. Now, okay, at this point, we had a phenotype, certain genes to suggest and certain pathways as a mechanism, but we wanted to see how these genes behaved actually from the cortices, from the actual experiments, in vivo experiments. So what we did is actually take the cortices from the in vivo experiments and run a nanostring assay, which is another technique to assess the gene expression. So we tested these 111 genes that we found in the RNA-seq. And what we saw is actually that only 2 of these genes were significantly deregulated, well, significantly differentially regulated in the cortex. But what was also important was that several genes that we already found upregulated in vitro seemed also to be upregulated in vivo. And these 2 genes were also found to be downregulated in vitro, as well as in vivo. So why am I showing these genes? Because basically there is more proof. There are indications basically that these genes might be a good candidate to continue researching the downstream mechanisms of 709, regulated pathways. So the same question popped up here. What if we don't compare these genes individually, but we try to find pathways that these genes participate in? And basically this is the analysis, the corresponding analysis. What I would like to keep in mind is that the only pathway that was downregulated, let's say, the genes in this pathway that was downregulated was ion transport. So genes associated with ion transport were again downregulated in the cortex of the in vivo experiment. And one of the highly upregulated pathways were the regulation, so genes associated with the regulation of excitatory postsynaptic potential. So again and again, genes that had to do with electrical activity were downregulated, whereas genes that had to do with energy metabolism and regulation of neural excitability were upregulated after 709 inhibition. So this is exactly where we are now. And this, for now, is the end of this project. Having shown that microRNAs both affect and are affected by the sleep homeostatic mechanism in mice, 709 regulates genes involved in the electrical activity, potentially via regulating energy metabolism of the cell. And as we saw in the in vivo experiment, the first in vivo experiment, microRNA 709 links neural excitability during wakefulness to the recovery process during sleep. And we have a theory which should be tested in the future, possibly with someone else, that 709 might actually have a protective role for the neuron, regulating processes that might exhaust the neuron itself. So this is basically our model. Sleep loss would upregulate 709, which would downregulate its targets, and therefore controlling energy metabolism and electrical activity. So that was the first project. Now the second project is kind of a cooler project and much easier to explain. So this project is called Rocking Mice, a model for the effects of rocking on sleep. And basically, as I said before, we've all sensed some type of this motion, either as babies or in the train or in the car, or in any moving vehicle that can cause a rhythmic, maybe, type of motion. So, however, in 2011 was the first successful study on rocking, on the effects of rocking on sleep in adults. And this was done by our collaborators in Geneva, where they showed that in a nap, with a nap protocol, so 45 minutes of nap during the day, when you rock people at a very low frequency, 0.25 hertz, laterally, people sleep faster, fall asleep faster, they sleep longer, and the electrophysiological markers of good quality of sleep are increased. So the question is, how does this happen? And that was our question, basically. To answer this question, we used exactly this type of platform. I was very accurate in that. Which also moves laterally in different frequencies, but the same amplitude. So we used 4 different frequencies because we first had to find out which is the right frequency for the mouse. And we started with 0.25, like in humans. We continued with 0.5, 1 hertz, and 1.5 hertz. We only rocked them during the light period because, as I forgot to say before, mice sleep more during the light period in comparison to humans who sleep in the dark. So we rocked them for 12 hours. This is the first observation we had. So in blue you can see first of all, these are hourly values of sleep, so how much time did they spend every hour in non-REM sleep. And in gray it's the average of the stationary baseline, so no moving, no rocking at all. And here you can see in blue the response at 0.25 hertz rocking, in green 0.5, orange 1, and dark red 1.5. And I hope you can also see that the last 2 frequencies, the faster frequencies, actually had an effect. And this effect, especially in the case of 1 hertz, was mostly in the beginning, so in the first hours after rocking onset. So we also wanted to assess what the total amount, if the total amount of sleep was affected. So here you see the difference between rocking days and baseline days. And as we can see, during the slower frequencies, the lower frequencies, there is no effect. But at 1 hertz, there is already a gain, so mice sleep more for about 40 minutes, 45 minutes more. When you rock them at 1.5 hertz, they sleep around 80 minutes more. However, whereas in the first 3 frequencies they don't lose any REM sleep, actually at 1.5 hertz they lose quite a significant amount of REM sleep. So about 10 to 12 minutes. We also assessed what we talked about before, the EG correlate of good quality sleep, so delta power. And although in the first 3 frequencies there was no effect on delta power, at 1.5 hertz there was actually decrease. Therefore, although you had a higher gain at 1.5 hertz, it was not the optimal frequency because they would lose REM sleep and the quality of sleep was not that good. So we continued our experiments and analysis with 1 hertz because this is what according to our observations, was the optimal frequency of rocking for mice. Now, we wondered, basically, how does sleep look like? How does each state look like when mice are rocked? And in this case, we measured the episode length and the episode number. What we saw was basically that while the episode length of wakefulness was reduced, the episode number was increased. In non-REM sleep, there was not a significant effect on the duration of non-REM sleep. However, the episode number also went up. And at REM sleep, we had no effect at all, as expected. So what that tells us is already that mice cannot stay awake for the same period when they're being rocked. And that was also supported by evidence on their sleep onset latency. So how long does it take for them to fall asleep when they're being rocked versus when they're not being rocked? So the latency was reduced in half. So the conclusion from this slide would be that rocking increases transitions from wakefulness to non-REM sleep, but not from non-REM sleep to REM sleep. Now, the next thing we had to answer was, how does this happen? What is the mechanism? And here I have to introduce to you your vestibular system. So basically, in your inner ear, you have an organ that can sense the balance, so that helps you balance, can sense gravity, can sense all types of acceleration of your head. So this organ is called the vestibular system. And no, actually this system is called the vestibular system, and it contains different organs. So the three canals that you see here, these three structures are called semicircular canals, and they encode angular acceleration. However, remember that our platform was going like this. That means there was no angular acceleration. Our model was strictly linear acceleration. Therefore, we were interested more in the organs that encode linear acceleration. And these organs are the utricle and the saccule. These organs contain these kind of structures inside them. And this is exactly how they function and how they're able to encode linear acceleration. So these purple crystals on the top, these are calcium carbonate crystals. And they form basically a platform, if you like, on top of these hair cells. Now, every time you accelerate towards the direction, this platform would move due to inertia. And every time that happens, the hair cells send a signal to the deeper structures, to the vestibular nuclei, basically. So this is exactly what would happen in this case. And this is what we think is happening to our mice when we rock them like this. Therefore, the perfect model to test whether this is the responsible organ that translates rocking effects on sleep were these mice. So these mice have a spontaneous mutation that does not allow them to have this platform, if you'd like. So they do not form functional otolithic organs. An otolithic organ is basically this platform that I was talking about. Therefore, they're not able to translate linear acceleration, and they have orientation deficits during swim tests, which means they don't know which way is up and which way is down. And they always carry their heads tilted. So this is look from a microscope, basically, of what I was just telling you. So mice with functional otolithic organs, they do have this structure of calcium carbonate crystals. However, their litter mates that carry the mutation, and they're called tilt-tilt, they do not have it at all. Whereas they do have everything else perfectly physiological. But the fact that they lack this platform means that they cannot encode linear acceleration. So we rocked these mice at 1 Hertz. We had all three genotypes, so the wild-type mice, the heterozygous mice, carrying half of the physiological information and half of the mutated one, and the tilted mice with non-functional otoliths. So in blue, you can see the wild-type mice, and they perfectly recapitulate whatever we saw before in the physiological mice, meaning a non-REM sleep gain of around 40 minutes, nothing significant in the reduction of REM sleep. Again, reduction in episode length of wakefulness, but increase in episode number, and so on and so forth, as well as lesser sleep onset latency. So that was also the case with the mice that had half the physiological information. However, their litter mates that carried the mutation, they showed no response to rocking, none whatsoever. Therefore, indeed, these organs were are involved in the mediation of the rocking effect on sleep. So, during a trip to a conference, basically, I bumped onto this paper, and this paper is from '73. And although I don't exactly agree, so what it shows here, it's a study on infants and the effects of vestibular stimulation, again, on infants, of rocking on infants. What it shows is that it's not the frequency, but it's actually the acceleration that is important. So, and you have to pay attention to. Although I don't exactly agree with the protocol they used, it gave the idea that don't focus on the frequency. It's not the 1 Hertz necessarily, but it's the achieved acceleration that is applied on the head that mediates these effects. So I went back to my high school physics, I guess, and basically in the equation for simple harmonic motion. And I saw that indeed, so if you change frequency and keep the amplitude constant, you also change linear acceleration. Now, for me, I ran to Paul and I said that it's not the frequency, it's acceleration. It's really what happened, by the way. And some months later, the answer to this by reviewer number 1 was no, you haven't shown that yet. Why? Because basically we did not, it's still possible, we did not show that the frequency is not important, right? So we did this experiment, which was the last experiment of my PhD, hopefully, with the tremendous help of Jan, because he had to build another platform that this time, which was not commercially available, that this time you could modify the amplitude. So we kept the frequency constant this time, we modified the amplitude, and we tried to achieve similar accelerations, or actually the same accelerations, as before in the previous experiment. So, and this is what actually I'm showing here. And I'm showing only the gain in non-REM and REM sleep, which was our first observation and the strongest observation, basically. So in blue, it's actually what we saw again from the first experiment, changing every time changing the frequency, 0.25 Hertz, 0.5 Hertz, 1 and 1.5. So in light blue, you see the non-REM sleep. In dark blue, you see the REM sleep. Nothing significant, nothing significant. Now, in green, you can see the results from the last experiment. And in this case, we try to achieve, we try to have as low an amplitude as possible, achieving a sub-threshold acceleration, basically. Whereas here, we try to replicate what we found before. And in this case, we tried to achieve, and we did achieve the same acceleration as at 1.5 Hertz, which, as you remember, had a larger non-REM gain, but had also a loss at REM sleep. And I hope that you can appreciate that actually we succeeded in, with Jan's new platform, we succeeded in replicating the results at the same frequency and amplitude. At sub-threshold accelerations, there was no effect whatsoever, even with the optimal frequency. Whereas at the highest acceleration that we could achieve, we always had a larger gain in non-REM sleep, but a loss in REM sleep. Therefore, the effects of rocking are, at least in the beginning, in the first part, mediated by linear acceleration. And as a recap, this is our working model, and I was very happy that we were able to show that rocking, although not expected by the way, we could show that rocking affects sleep in a rate and time-dependent manner. We could show that it favors transitions from wakefulness to non-REM sleep, and that the effects of rocking on sleep are mediated by the applied linear acceleration encoded by the otolithic organs of the vestibular system. So, just as a final slide, I want to give some relevance because they might have seemed too basic. These projects might have seemed too basic. For me, trying to see the bigger picture, so microRNA research, as I said, still has a lot of field to cover in the sleep field. It's quite new, and I think it has a lot to offer. We are at the point now that already there is research, there are studies for microRNA signature of sleep disorders, and in other fields like cancer or like the cancer research or neurodegenerative disorders, there are already patents or clinical trials, so inhibitors in clinical trials, although not very successful so far, but still there are. So before we go to something like this, it's very important to understand the functional role of each microRNA in sleep. And when it comes to the second chapter, well, this is more of a perspective. I would be very happy to see further studies in follow-up studies in deciphering which deeper structures actually are downstream of the vestibular organs, the vestibular nuclei, and receive this kind of stimulus. And this would be very essential to create non-pharmacological applications, which are already, there are efforts actually to achieve them, to construct them. Also for instance, for relaxation in the elderly, as well as relaxation in university students, for instance, as a study from 2014 had. So with that, I'd like to give an end to your torment, and I would definitely like to thank the Paul Franklin Group, past, present, and future, and definitely the GTF, Johan, Hannes, Corinne, Leonor. Well, very much I would like to thank Christos Polysopoulos, because he helped me out. We found this analysis, WGC analysis together, actually over beers, trying to answer Paul's question. Our collaborators in Geneva, with a big applause for Aurore and Laurence. Our collaborator in Montreal, Valérie Mongrain, and the Sofia Fenduli Foundation that supported us for the in vitro study in the microRNA project. And definitely you for your attention.
It's time for the experts and the presidents to have a chance to. Yes. So once again, in front of everybody, if you wish to say a few words and eventually ask questions, please don't have.
Yes, so first of all, Kostas, I would like to congratulate you for your very nice work. It was very nice to follow up on what you did, talking with you at conferences, interacting with you during the exam, and now hopefully I have the honor again. And yeah, it was really nice to see
So, if I understood the question correctly, first of all, yes, we came across 709. To me, it makes more sense that more microRNAs regulate the same step. Now, putting that aside, microRNAs are called fine-tuners. Well, for me, if you ask me, they're part of epigenetics. If you ask Paul, they're not. But I stand by my opinion, and basically, basically, I think they're with their lifetime, they are faster and more capable of adjustment. So they offer basically another level of adjustment to the environment of the cell. They prime, if you would like, they prime the neuron for based on previous activity. So they prime the neuron to keep it alive, if you want, in this case. At least this is my speculation here. So again, I don't know if I understood exactly the question, but in general, this double regulation, why does it exist? I think it offers another way, another more flexible and faster way of regulation. This is why microRNAs in general exist. I think this is what gap they cover in gene regulation.
Well, I mean, could it be that the G-protein coupled mechanisms, they regulate that and they might be part of the 709 expression as well, no? Because it's an activity-dependent gene. So it could be just one of the pathways that is targeted by rate-promoting compounds, no? By rate-promoting the neurotransmitters, like noradrenaline or serotonin.
Yeah, I mean, in a lot of cases, you see also that microRNAs serve feedback loops, negative feedback loops, for instance. Again, to sustain balance. That is to put a ceiling, if you want, into processes that might damage in the end the cell.
And then my second question was related to the second project, if I may. In my experience, it's always good to choose a species for testing certain behavioral effects in relation to what they do in their natural environment, how they develop and how they function. And for me, rocking is something that is very innate to species where you have prematurely born offspring. No? The rocking is important because they are attached to the mother, they're carried around by the mother, so there the rocking could be very useful. But for mice, why do you think they're...
This is a very good question, and well, so I don't have a confirmed answer. I have a theory though, since the first year of the PhD. So the vestibular organs, both in humans and in mice, in rodents, they develop very early, embryonic stage 17, if I remember correctly, in a mouse. And they're there, and they actually can translate linear acceleration already from the belly of the mother. So evolutionary, or developmentally wise, if you look at it, it does make sense that it's a familiar motion, a sensation, basically. It also can, I mean, we did not test this, we did not prove this definitely, but we also did not test this, whether there is a relaxation effect downstream. So I could say, for instance, I know that there is this theory from other groups working on in humans in the same subject, that this is the actual effect of rocking. It relaxes, and this could also have a sense, basically, in the development of embryos or premature babies.
Okay, thanks a lot.
Thank you.
Thank
So
you.
next in order, Paul, please.
Sure.
So congratulations from me for a very nice and very long talk.
I took my time.
I have some questions for the difficult part, this first part, right? Like it's so confusing to me that one microRNA can target so many, and one gene can be targeted by several microRNAs. And so very indirectly with the RNA-seq data, you try to infer what the targets are. But what would qualify for you
Yeah,
evidence
correct.
still of what pathways are involved?
So there are techniques where you can, first of all, the first step would be to validate whether a specific target or some specific genes are direct targets of this microRNA. For instance, some candidates that were proposed by the different analyses. Now, beginning with that, there are techniques where you can fish out, let's say, the direct targets of a microRNA. Unfortunately, although there was, I think, in the past discussion, or at least I thought about using them, the technique was not fully validated, basically. So that, to me, would be one first step to really be able to capture, to precipitate the targets with the microRNA itself. Another thing that could be done was also, I mean, now we inhibited the microRNA, we could go the other way around. Although this approach is not always the best, overexpression of the microRNA, still, it would prove, if the results would be positive, it would prove better, it would support actually the idea that a gene is a direct target of 709. Now, even more, if the this overexpression would be by injection in the brain, and by some chance we saw exactly the opposite, we could also confirm that it's a very specific relationship between the phenotype. I mean, I know this was not your question, but we could also, yes, validate that this is a specific relationship between the microRNA and the phenotype that we observed. But again, there are many pitfalls in these techniques. So I would still go with the precipitating one, or even we could use. Let me think a bit. There is also luciferase assay that would be used in this case. But yeah, so there are several steps. But what I'm trying to say is there are several steps that could confirm this. Again, that a gene is a direct target, but the next step after confirming this would be again to go to protein level and try to connect even further the phenotype with this.
So now that you have your PhD, would you put a new PhD student on this project? Do you think it's a good idea?
Actually, I would, because I do believe, as I said before, it's true. I believe in this, that there are a lot of information there. But I would not only focus on one microRNA. I would focus on several ones, trying to figure out, for instance, is there a combination? Is how each, so for instance, let's say we have 5 microRNAs. How does the phenotype change every time we have a different combination? Which is not one PhD, but I guess more, but still.
I saw you had a voltmeter there at some of your slides.
Yes.
So the EEG is an AC signal or a DC?
It was more to, it was more to say that it's a measure of power. But
But it was a DC.
it was very hard to think about that.
I have many more questions, but maybe we give opportunity to
large audience. Yes.
Okay, so given that this mRNA is affecting some fundamental functions of the neurons, do you know if they are also involving other behavior that you have not observed? For example, I saw that there is some expression in the hippocampus. So do you know if these mRNA and microRNAs are also involving, I don't know, learning and memory or other physiological?
Yes, so specifically for 709, right?
Yeah, well,
We're talking about.
yeah.
But so specifically for 709, there are around only 5 studies in the brain, and the only additional behavior that that actually has been confirmed is stress. So it was upregulated, it has been seen to be upregulated in the prefrontal cortex and the hippocampus after stress, in rats specifically. But other than that, so 709 is ubiquitously expressed. It's enriched in the brain, but it is nonetheless ubiquitously expressed in the whole organism. So there have been other studies showing that it does affect the cell cycle somehow. It does regulate expression of other microRNAs, 15 and 16A. So yes, now to turn this around, there was a study in 2016 that specifically about 709, I mean, also trying to make a point for the rest of microRNAs, that it does make, it's of essence if, what the tissue is. Sorry, I'm trying to think. So it's really necessary to know which tissue the microRNA is expressed in, and it's not enough that the gene and the microRNA are expressed in any tissue. What I'm trying to say again is, maybe in adipose tissue, 709 targets GSK3 beta, which it does. In the neurons, it doesn't, or in other tissues. So that was the point of that study. Trying to say again that it might be totally different, even if it's expressed in the hippocampus, for instance, and if in comparison to its expression in the cortex, the function might be totally different. The targeted pathways might be totally different. I didn't get the question. You had a question about those people, but I didn't.
Yeah. Are people with any inner ear disorders insensitive to rocking?
Oh. So far, so this is a very good question. I think, so there is no study, definitely no study about that. But I think they might, no, never mind. There are thoughts anyways to go towards that direction as well. From other groups working with humans. So I don't know how to answer. I would guess so. It depends actually, because with a labyrinth, it's a worse situation. It's this is just a small mutation which just vanishes this structure, the otolith, basically. With any other kind of disorder, it might have additional problems. That's the issue, because you have more systems that are affected, or more parts of the same system that are affected as well. This is why we were very lucky with this model, because any other model had to do with destruction of the labyrinth, destruction of other organs at the same space, basically. So yes, it might, but maybe there would be also other factors there.
So this is maybe a little bit speculative, but you had a molecular and a sensory approach. Is there any way of seeing whether the two are connected to each other? So for example, the genes that are dysregulated in the molecular approach, do you find them also being maybe dysregulated in knockout mice that they have of the defect of the sensory organ?
Okay, that was very specific. So I know of studies that are searching for microRNAs in the vestibular system for differential expression or differential regulation of microRNAs in the vestibular system. But I guess when it comes to excitatory, so neural excitation or excitability, I guess there would be some overlap, let's say, between the genes that we find being targeted here and the genes that might be upregulated, or the pathways even better, that might be triggered during rocking, for instance, because of the hair cells being excited by the stimulus and so on and so forth. But there was the idea in the beginning, there was the idea to also assess gene expression in the vestibular project, in the rocking project, but that didn't take place.
Not sure that I get really what you said about the microRNA, but you said fine-tuning?
Fine-tuning, yes.
And also a fast process. Right?
Yes.
Okay, so can you try to tell me why the sleep, the long process, that we don't need really
So
We need this fast response.
my first word that popped into my head would be accumulative. So it's possible that for the level of the cell, it has to be a fast process, and it also is very likely that it's a process that participates in other behaviors as well. But in the level of our brains, basically, it doesn't need to be a fast process necessarily. It can be accumulative. For instance, the upregulation of 7 or 9, right? It can be accumulative. It can be fast for the cell if it needs to. No, I see now your question. Give me a second.
What is fast, what is slow?
Yeah. Isn't it possible that it can do both? Because the fact that it can, when you say that something is fast, it can mean that it's created fast, produced fastly, and the turnover afterwards could be also fast, or one of the two is fast. So in general, for microRNAs, you can have both. You can have also relatively slow, you can have fast because of the turnover, you can have fast because of the production. You can have, what I'm again trying to say is that you do not have to have a slow microRNA for a slow process in the end. You can have a fast molecule that, however, the processes that make it fast can be modified depending on every time the environment. This is what I'm saying. This is what I'm thinking about. Sorry for the long way to that.
If not now, you have also Konstantinos available afterwards for additional questions. We have a little retreat to do. A short one, fast one.
Όχι ρε, αράχτη. Ευχαριστώ πολύ που είστε όλοι εκεί. Thanks.
Okay. So, Konstantinos, we are happy to tell you that we are going to recommend to the rectorship to award you the PhD in neuroscience from the University of Lausanne and Geneva.
Very good.
Since it is a joint. So, congratulations. And so this is a temporary imprimatur that suggests you accomplished everything. But there's still a little path to see with the map and a few pages of instructions
Very good.
until you may eventually receive the diploma. Don't follow them well.
I will try. I'll do my best. Thank you. So, okay. Can I say a few words or yes? Maybe?
Sure.
Okay. So now it's the hard part, and so thank you very much for being here. First of all, many thanks to my committee, because all the interactions we had were very nice, very supportive all the time. I would like to thank basically all of the people of the CIG, because these 5 years have been, well, a blast, I guess. No, they have been very nice, some of the best years of my life. So, and definitely the people, all the people here, from the GTF, from the phenotypage, from the animal facility, everyone from the CIG made this happen, basically, with their smiles, the good mornings, and all the interactions. So that's one. Now, I definitely would like to thank all of the guys in the TGIF group. So all of the PhDs and postdocs, basically, of the CIG, because they have been an amazing company, and I have not been one amazing company to them, but still, they're there, and they're very supportive. So thank you very much for that. I would like to thank, well, the Paul Franken group for sure, the lab, you know, Shanaz, for instance, Maxime, no, Geraldine. Well, I can't say why. I mean, Geraldine for being my mentor, Shanaz for being very joyful, I guess. Yeah, Sonia, well, for everything. Cool, for being cool, right? Jan for teaching me how to fish, definitely, one time. Maxime for just whispering me to once this thing about hub genes, and then everything evolved with analysis, but also for being very cool. Yeah, so anyway, long story short, everybody contributed to me having a very nice time here. Thank you very much, Paul, for giving me the opportunity to do neuroscience when nobody else would, you know. Well, I guess. Now I can tell you, right? After 5 years and a PhD. No, I mean, I'm gonna say the same things that Marike said last week. You're a very, yeah, no. You are to blame. No, you are a wonderful person, and
Don't embarrass me.
yeah, I try. A fine, no, a very fine scientist. So really, thank you for teaching me how to do proper science, and very accurate and painstaking. You know, writing papers, for instance, reanalyzing stuff.
Painful.
So yeah, thank you for the opportunity. Thank you for trusting me. Thank you for trusting my ideas. Definitely, I'm gonna look for any employer in the future that does at least that gives me some space to work, as I learned here with you. So thank you very much for that. It's, yeah. So Marike and Jeff. Hi. So I would never have imagined that I would have such colleagues, basically, never. And I would never have imagined, well, I mean, the amount of talking that Jeff does, for instance. And the times that we have disagreed with Marike, I mean, that's, yeah, in 5 years. So thank you very much for all of this. Thanks for keep on talking until I put 1% of knowledge in my head. And thanks for disagreeing with me and making me a better scientist as well. Thanks a lot. Also thanks for the ping pong, Kan and Jan and Alessandro, if you're there. And Jeff, that was also a very nice time. So, okay, to continue, a thank you to my parents. Hard. Look at that. Thank you. So, no, thank you for being there. Thank you for being so supportive. Thank you for, well, preparing all this food, basically, that I'm keeping all these people from eating. No, thank you for being like very strong and the strongest people I know, very cool parents, and very good friends as well. And difficult again, look at that. Thank you very much, Maria, for, well, I mean, everybody who knows me at least once must have heard the name Maria. So thank you for being there all these years. Again, thanks for all the advice. Thanks for, I don't know, pulling my head up when it was too much, I don't know, in work or couldn't see the light and showing me what life is. And yeah, thank you for being there because none of this would, you know, make sense, basically. Yeah. Thank you guys from Zurich, by the way, for being here. It's really an honor for me. Thank you wherever you are over there. So my friends from Greece, from 10 years old, from 18 years old, from Thessaloniki, maybe in Frankfurt, Brussels, wherever you are, thank you very much for everything, for putting up with me, for being there for me, and for wanting me to be there for you, even though we're far away from each other. So, thanks. And last but not least, all the guys in Zurich, thanks for tuning in, you know, tune with pun with the title. Thanks for tuning in to hear me talk, and I hope you didn't die of boredom. I'll see you soon. So, thanks. So, P.S., thank you, Iris. You have been wonderful. Thank you.
Very short, two words to thank Kostas. I find it a bit sad, you know. 5 years is a long time. Also, Marike did presented her thesis last week, so maybe they should shorten these PhDs to 3 years or so, that you don't get attached to these students so much. Yeah. So this was 5 years ago that I interviewed you over the phone, and you were very helpful and polite and corrected me and helped me say, when I tried to say Konstantinos and it didn't come out, he said, "Oh, you can call me Kostas," you know. And this is him, you know, very polite, very helpful, and so forth. It's maybe a bit boring, right? Nothing special to say about things that happened or no. I only learned that Kostas does better after a couple of wines in presenting his research, so maybe that yeah, would have helped. For sure. That's how you also won this poster award at one of these sleep meetings that we had here in Lugano, I remember. So, yeah, otherwise, thank you for all the good work that you did. And everybody loves Kostas. That's thank you. What do we get now?
Special treat. Can you turn the thing up? Wait. Somebody knows how this works? The sound?
The bottom of this.
The sound? Does somebody know or not?
Kostas,
He's very helpful.
Yeah.
sound.
Sound, yeah.
Do you maybe the panel is over there?
What? Here?
No.
There.
It's
It's volume. Here.
Voila.
Is there?
Let's try.
How much?
Oh, is it
you have to change the input.
display on off? Yeah, VGA. I think.
Yep.
Oh, so it's an organized desktop. Yes.
So I would like to thank the organizers for giving me this unique opportunity to present our work actually. That Maria K is gonna introduce.
Dr. Kaan for this very kind introduction. And again, thanks to the organizers. I know it has been a very long day, but we have such exciting data that we have to present it to you. So today, Kaan and I are going to talk about Dr. Kostas Thessalonikos, or also known under the common name Malaka, a new discovered species. And actually this exciting data and conclusions has recently been accepted for publication in the Journal of Insignificant Results. So without further ado, we're moving on to a bit of history. So actually the first footage of this species was taken in Lausanne in December 2015. Or actually we think that this is our Dr. Kostas, but we're not completely sure. So we started to look a bit more on the internet to see what is known already about the species.
Yeah, actually, there were some rumors about it. Like some villagers told us, like we've seen this something, you know, like out of like nowhere. But they were positive that it was in Lausanne, and we were very excited because this is like a species that nobody worked on so far, and we are like, okay, let's study that. And what you do? The first thing, you go to Wikipedia.
Of course.
And one of the very exciting thing in science, like you put something to Wikipedia and you don't find it, you are like, oh my god, this is like an ocean, you know, like nobody worked on that. We can just, you know, like publish tons of papers and
Exactly. It's a
have
new
our
hot
own
thing.
laboratory, you know, like hire some PhDs,
Exactly.
postdocs and do research on
Let them work their
all
asses off.
this. Well, the second thing that we ask is where he's coming from, because we never seen him in Lausanne or Switzerland. Well, previous studies actually showed Kostas-like species were initially found actually this region around Greece, and there are also some studies show that he was seen in Netherlands, but nobody's seen him in Switzerland. So that was very exciting, and then we decided, okay, Marike, we're gonna do it.
Yes, so we're gonna dive a little bit deeper into this like migration path that this special specimen took. So he migrated basically from the Mediterranean shores to the Alpine mountains. Why? What's going on there? What kind of hints can we get from this pattern?
And it's like a species in two extreme conditions. I think it's over 40 degrees or something.
Yeah,
He's
it's very
burning
strange.
out there, and it's like freezing cold.
Very, very strange. But we did notice something. He likes to appear in the proximity of alcohol. And it seems that this is a pattern that is like reoccurring. So here there are two other images of our favorite specimen in the neighborhood of alcohol again. So it seems to be important. So can we gather more data about this Dr. Kostas Thessalonikos in close proximity with alcohol?
I mean, we know that this species has always alcohol, but we have no idea what alcohol does this species. So, and we just gave him different alcohol, let's say. We gave him like rum and coke, one of his favorite, like some different beers, and we just like observed the behaviors. But I think these studies were inconclusive. We were not understanding anything because every time he was just showing a different sort of behavior.
Yeah.
So we couldn't conclude what was going on.
Very difficult.
So what a biologist do actually to understand the function of something, for example, you make a mutant, you know, and we don't know about his gene, we cannot CRISPR him out or whatever. So we decided that, okay, we start like very easy. Like we start by taking the alcohol away from him and see the effects, like how long a Kostas will survive without drinking something. And then, I mean, it was difficult.
It's very difficult.
Like, I mean, we designed the experiment that we were going to make him alcohol deprived for like a week. But actually, at the end of the second day, he was suffering so much. And I mean, we were unable to like,
yeah, we have to
proceed.
stop it.
It was
too
against,
early. Yeah.
what convention was that? Marta, you know it the best. Yes, so it was against ethics. We couldn't move on.
He couldn't survive.
Yeah,
Yeah.
and then we just said like, okay, our specimen is like very precious. We just keep it here, but we can conclude that he kind of needs alcohol for like proper functioning, let's say. And then the second question, of course, like we have to understand what kind of nutrients this species needs for proper development and function. And we understood like it's mainly hunting, but occasionally gathering. But this specimen stated so many times that he's a foodie. I don't know, is there something called foodie? But he says there is, and he is one. So even though he has high preference to red meat, and he likes barbecue and everything, and then for him, this is like a sign of domination. He has to be the one making the barbecue and eating from the barbecue directly. Well, but we couldn't find like something significant. He is eating everything.
Yeah, again, the pattern is very difficult. So I think a very interesting and striking phenotype that appeared from this specimen is sleep-wake behavior. Actually, we have never seen something like this before. It is as if there is no sleep homeostat. This specimen can sleep anytime, anywhere, on one hand. But then on the other hand, it can also push itself to stay awake until it has finished whatever it needed to finish. This is amazing, and I think this species has like a lot of potential for new breakthroughs in sleep science, because let's face it, we still don't know why we sleep, right? So what we did is we collected some data from
Real data.
from some Homo sapiens, subject A and subject B, so what you can see here. So this is some normal data, and I would like you to focus here on the gray areas. So, I mean, this kind of looks normal, right? But then we looked at our Kostas. My God! In the gray area where he's sleeping, there's no brain activity, nothing. Is he like super efficient? He's like extremely well recovering during sleep. Is there maybe, so there are like rumors, but they're not confirmed yet, that he actually has like this rocking bed. And this is why he sleeps so efficiently and he can be so much awake during the day. But we need to perform further studies to see if this is actually true. Let's see.
Well, besides the sleep, of course, we look at other type of behaviors that this Kostas species, let's say, is exhibiting. So I want to talk about the courtship display. So here is a beautiful peacock, and this is a male, and they are trying to attract the potential mates. And the courtship display is a set of display behaviors in which an animal attempts to attract a mate. These behaviors often include ritualized movement, dance, vocalizations, or displays of beauty and strength. So we later want to see if he is performing any courtship behavior display, and then we further analyze. And actually, like, we found one that he is doing a lot of these ritualized movement, like dances, sometimes individually, sometimes together with a partner. And he also has...
We have another footage of him displaying this courtship
this
behavior.
very weird dance, like we never seen it, with an umbrella.
We think it's courtship behavior. We're still not completely sure what the exact function of this behavior is. But yeah, and and of course we also have the sophisticated expression of yeah, we think it's beauty. I mean, the tears of seduction are literally rolling down his face, right? So yeah, it's clear. And then of course the last important male behavior to express is strength, and you can really see it here. He's like this alpha male with goggles, because that's very important if you're Dr. Kostas Thessalonikis.
So given that there are so many courtship display behaviors that's performed by this individual, we wanted to analyze and understand its fitness and how he will survive and find a mate. And the previous studies were actually quite demotivating, given that a lot of attempts were failed. Like, you know, like he was rejected by the other party most of the time. But we didn't give up. We continue observing, we continue experimenting until we found that. So he doesn't have to perform any of this beauty, strength, or anything else, but he just have to do this mimicry, you know, like whatever this person is doing, like he's doing the same thing, and then voila, he's successful.
Yep. So basically, we describe now a couple of important features, I think, of this newly discovered species. So we've seen where it comes from and where it's living right now. We've seen some specifics about its liquid and food consumption. A very striking sleep phenotype, which I think definitely deserves more attention. And this very complex courtship behavior, which indicates that there's also a lot of neuronal activity, I think, going on during waking.
More studies will follow on you.
Yeah.
Yeah.
But we had some additional observations that we didn't really know where to group them, but we want to share them with you because we think it's of importance to see how narrow-minded Homo sapiens sometimes can be and how we have to try to think out of the box. So one of these observations was that this specimen said cars with automatic transmission should be driven with 2 feet. So we were like, okay. So let me remind the audience. The upper part of the picture represents manual transmission. So normally you would drive with your left feet, you would use the clutch, and with your right feet you would use the brake and accelerator. So this is a car that you find a lot in Europe. On the bottom part, you can see a car with automatic transmission. This is a car that you find often in the United States. So you could imagine that if you use both your feet, so you use your left feet to brake and your right feet to accelerate, I mean, it's difficult, right? It's,
Don't try.
I mean, I'm too narrow-minded. I didn't dare to try. But this specimen, he's so amazing. He thought out of the box and he tried. And we almost died, but we didn't, fortunately. So.
How long it took to convince him about this? Like, did he, you know, like, I'm wondering, like, did he learn this?
Yeah, so this behavioral flexibility it's not so, it takes some time. I think we call it stubborn. Yeah, exactly. But after 2.5 hours, he figured it out and we were all fine.
Well, like, we continue our observations and there are so many. So, like, I remember a night that we were out with Kostas and some person that I don't want to say his name, not to humiliate. Hiya. And so, one of us in this story was drunk, okay? So that guy is not having the conversation, but this specimen and we were chatting how to make him better, you know? And I was like, dude, you have to drink more water to feel better the next day. And he was like, no, you should not drink water. You should never drink water after alcohol, you know? Like, that makes you worse. Like, the worst hangover that I ever had was drinking water after. And we didn't believe him, and we experimented him directly. And the day after, we just gave him a lot of alcohol, no water. This is the result, serious result. He couldn't wake up. This is a Kostas, just like a friend.
Not so good. But we still need a control experiment, right?
Yeah.
Because we still need to have add the water. But yeah, obviously.
And Kostas, this guy is a man of like big words, you know? He can go something like, I have been stressed twice in my life. Okay, so just to update that, were you stressed before today? A little bit? You were in emergency?
Just a bit, yeah.
Ah.
We have to update.
Yeah, we have to. Sorry, it's a bit outdated.
Well, okay, and
okay.
we asked, okay, when you were stressed. And it's funny though. And he mentioned that we like to play ping pong. And once he was very stressed because we were playing ping pong in the lab. And he was so scared that like he would break something. So he was stressed because of that. The second reason that he was stressed, you know what happens when you lose in ping pong? You buy a kebab. And indeed, he bought me a kebab for that.
So there's this there's this recurring phrase that this specimen mentions very often. Next stop, my DCK.
What's DCK?
Yeah, we didn't know for so long. It's so puzzling. So in the end, we're biologists and not mathematicians, so we hired a mathematician. And he ran all kinds of complex models, algorithms, like
brilliant guy.
cannot explain it to
Amazing,
you,
brilliant
don't
guy.
understand it anyways. But the outcome, we were like, of course. We could have thought about it. Duck, next stop my duck.
So this new species is very naive and he's thinking out of the box. It's
Yeah.
amazing.
We can learn so much from it, really.
So we have, as you see, a lot of data and we submit, like collected everything, write that, and then we submit that, and of course we got some reviews. And the first reviewer, actually, like both reviews were positive, but this was more critical. Like this paper describes the phenotype of a novel species, Dr. Kostas Thessalonikius, or whatever, which has high potential to understand better phenomena in biology, such as sleep. However, it does not explicitly explain how such a peculiar species could survive, given this self-destructive behavior. We strongly suggest that the authors address this in more detail. And the second reviewer, it was fine. Best paper ever.
So we went a little bit more into the comments of the first reviewer, because we thought, yeah, it actually makes sense that there must be something to him that makes him able to survive, right? Because otherwise he would have already died of alcohol intoxication.
Water.
Or water, yeah, water. So we had a closer look at his professional achievements, and then we actually started to realize that he's a very successful scientist, that he's a great colleague who shows a lot of support during the day and also during the night when necessary, that he has a lot of critical thinking that I really appreciated, and I think the other co-workers in the lab as well. And last but not least, as a proof actually of all these abilities, he was awarded last month the amazing presentation at an international conference with an audience of 1,800 people, which I think is worth an applause. This is one way to compensate for the previous behavior, we think, but it's still an hypothesis, right? Huh? It's a suggestion. I think the second thing that's how he's compensating is by being an awesome friend. So I think that if somebody's stressed, this Kostas is the first to know, and you sometimes know it before the person itself knows it. And when you realize you're always there, you're helping, supporting, talking, and that's really great. I think you're a great friend. So thank you for being around. And I would like to hand over to my colleague.
Well, this is the hard part because making fun of somebody is always easy, but even good verse is difficult. But Kostas is an interesting person, and Kostas, I directly speak to you right now. So I like you a lot. One of the reasons that you are competitive, oh my God, you can play ping pong all day until you win. And then he can play poker all night until he wins. And there's, we were one night at Steve's place, everybody was out, we have to work the next day, and it was only Kostas and I, two people were playing poker. He was getting my money, I was getting back the money, at some point we were like, okay, we have to quit. So I'm also competitive, but thanks for being very competitive and challenge me. The second thing that he likes challenges and he accepts all of them, and he does try a lot of things, and he's always good when he tries. He can make very boring things like a repair work at home fun. So he's also, I guess, enjoyed very much the life in Switzerland by fishing, relaxing, and everything. And he's always surrounded by a lot of friends, a lot of you that always with him, which is really a great personality, I guess. And one more thing before I finish. A lot of people think that you're 50% rum, 50% cola. I really think that you're 100% pura, malaka. And I really want to say Siharietra, yeah. And congrats.
Yes. You're cordially invited to the apéro. Unless you want me to present again. Honestly? Yes, again. Yeah.
Right, you didn't put it here so it wasn't on the screen.
So where was it?
Well, you should have put the presentation here. The stream was running here.